Hello everyone, and welcome to my presentation about analog experiments for the detection of bacterial biosignatures in ice grains from ocean worlds. Active ocean worlds are some of the most interesting solar system bodies for astrobiology. Saturn's moons Enceladus and Jupiter's moon Europa are predicted to harbor global liquid water oceans beneath their icy crusts. In 2005, the Cassini spacecraft discovered plumes originating from the South Pole region of Enceladus from four main surface fissures, called the tiger stripes. These plumes are composed of vapor and water ice, and they eject ice grains from the subsurface ocean into space. Some of these ice grains escape Enceladus' gravity and comprise Saturn's diffuse E-ring. Like Enceladus, Europa could be cryovolcanically active. Some recent papers reported evidence of plume activity. Sputtering and micrometeorite bombardment may also eject icy surface particles to high altitudes, where they can be sampled by spacecraft. Ice grains ejected from icy moons can be analyzed during spacecraft flybys by impact ionization mass spectrometers, such as the Cosmic Dust Analyzer CDA on board the Cassini spacecraft, or such as the Surface Dust Analyzer SUDA that will be on board the Europa Clipper mission. Impact ionization mass spectrometer record mass spectra of ions that are generated by high-velocity impact of ice grain into a metal target. This instrument can determine the composition of the ice grains and potentially indirectly sample subsurface oceans. In Saturn's system, data collected by Cassini's CDA instrument show that Enceladus ocean is salt-rich, sustains hydrothermal interaction, and contains a variety of organic material, including complex macromolecules and low-mass volatiles which may act as amino acid precursors. These discoveries were supported by laboratory analog data using LILBID, the laser-induced liquid beam ion desorption experiment that I will present in the next slide. On Europa, the subsurface ocean is also predicted to be in direct contact with silicates and possibly with seafloor magmatic activity, which would enhance the potential habitability of this moon. The upcoming Europa Clipper mission will be able to sample ice grains originating from Europa's surface or the subsurface ocean. Interpreting the data recorded in space required calibration via analog experiment. In space, impact ionization works in a way where the ice grains impact a metal target at high velocity. This creates ions that are then detected by a time-of-flight mass spectrometer. You can see here on the left the resulting mass spectra of water ice grain. The x-axis shows the mass of the detected molecules, while the y-axis shows the intensity. In the lab, we simulate the hypervelocity impact of the ice grains with the laser-induced liquid beam ion desorption experiment, or LILBID. Here, the impact ionization process is simulated by irradiating a micrometer-sized liquid water beam that contain dissolved analytes with an infrared laser. When the water beam absorbs the laser energy, it is heated up and it explodes into fragments. The created ions are then accelerated by a high voltage potential difference, and they are detected in a time-of-flight mass spectrometer. You can see here on the right the mass spectra of water, which is comparable to the previous spectra from water ice recorded in space. The LILBID experiment can be used to calibrate space-borne mass spectrometers to identify biosignatures that are potentially incorporated in ice grains. This approach has shown that bioessentials molecules, such as amino acids or fatty acids, for example, these molecules can be reliably identified in ice grains at concentrations as low as the micromolar or nanomolar level. Abiotic and biotic formation processes of these molecules can be distinguished from each other based on spectral features. So here I present the next steps of the LILBID experiments. We measured cell material extracted from cultured bacteria to predict their spectral appearances in impact ionization mass spectra in both cation and anion mode. So we isolated cell material from two different bacterial species, Escherichia coli and Sphinxopixis alaskensis, in short, E. coli and S. alaskensis. E. coli is a well-studied model bacterium, while S. alaskensis is a marine bacterium that was isolated from Alaska cold seawater. It's an oligotroph bacteria, so that means it can live with low nutrients and is very small inside. It's a very good analog bacteria to potential life forms on icy moon oceans. The material we extracted from these bacteria includes genomic DNA, membrane lipids, and the aqueous phase of the lipid extraction. These extracts represent potential biosignatures, and they were also investigated in sodium chloride-rich matrices to account for the salty oceans of the icy moons. We also investigated lysed cells of the bacteria without any extraction. Our results show that biosignatures from the two bacteria are clearly visible in both ion modes. Here you can see as an example an anion spectrum of DNA extracted from the S. alaskensis bacteria. We identified two nucleobases, adenine and guanine, and several nucleobase fragments. We also identified compounds deriving from the phosphate deoxyribose backbone of the DNA. We also extracted lipids from the two bacteria. Here you can see the spectra of lipids extracted from S. alaskensis, where we clearly identified many lipid fragments, such as deprotonated fatty acids. Here you can see the spectra of E. coli aqueous phase. So at the end of the lipid extraction, we obtain two phases. One phase contains the lipid, and the other phase, the aqueous phase, contains all the polar molecules. So here in the aqueous phase spectra on the right, we identified guanine again and some nucleobase fragments and many compounds from the phosphate deoxyribose backbone of the DNA and also several amino acids. We compared the lipid spectra between the two bacterial species. Fatty acids are especially interesting because their relative abundances can serve as a discriminator for biosignatures. In most organisms, fatty acids are produced by the addition of 2 carbon atoms at a time. So this results in an excess of fatty acids with an even number of carbon atoms, and also with C16 and C18 dominating. On the contrary, in the abiotic synthetic of carbon chains, the carbon atoms are added one at a time, so there is no difference between even or odd carbon number. So here, as expected for biological samples, the spectra from both bacteria show a predominance of even carbon number fatty acids over odd carbon number fatty acids. We also see that C16 and C18 fatty acids, are also C15 for S. alaskensis, are the most abundant fatty acids. We also noticed that the S. alaskensis bacteria contains more unsaturated fatty acids than E. coli, and this difference may be due to the different bacteria species, which may have different membrane composition. We investigated whole cells of S. alaskensis. So instead of performing a chemical extraction, we only lysed the cells by sonication. This process makes more sense than a chemical extraction if we compare to a potential detection of these compounds on missions such as Europa Clipper. Here you can see the spectra of the lysed cells in the positive mode, and we identified 2 nucleobases, adenine and thymine, many nucleobase fragments, many amino acids, and some sugar of the DNA backbone. In the negative mode, we also identified many lipids. I didn't show the spectra here because of lack of time, but if you are interested, I could, of course, provide more spectra. Finally, we also investigated DNA in salt-rich matrices. So here on the spectra, you can see that we observed a lower peak for the nucleobases and sugar than in pure water spectra. And overall, the spectral sensitivity of the extracts decreases with the increasing salt concentration. So for conclusion, our results show that microbial biosignatures can be identified with impact ionization mass spectrometer. And in particular, we observed nucleobases, the fragments of the phosphate deoxyribose DNA backbone, many amino acids and lipids. Ideally, a future applied instrument should be capable of detecting both cation and anion to detect the complete range of the biosignature that we have so far investigated. Our results complement a spectral reference library for Europa Clipper and other future ocean world missions. And ongoing and future work include the investigation of other microbial species, phages, and other potential biosignature. Thank you very much for your attention, and I would be happy to answer any question.